Let me introduce all the people that are coming up, almost all of which have spoken already today, so probably don't need much introduction. So sitting first is Jennifer Panagoulias, who you just met, who is the regulatory advisor for genetics, Dr. Timothy Yu, who is the PI for genetics biotherapeutics, Dr. Art Beaudet, who is obviously a world expert in genetics and also a physician who can answer a lot of questions about our children and has probably the most experience in Angelman syndrome on the table. Dr. Scott Dindot, who you just heard from, also a geneticist and an expert in the paternal imprinting defect. Dr. Jim Wilson, who is an expert in gene therapy. Meghan and Michelle from Roche, who are developing an ASO program and also have become experts in outcome measures and biomarkers, I think, by default, with all of their work there. Dr. Ed Weeber, who is working on a gene therapy program with PTC. Dave Segal, I think, is behind him, who is working on various different approaches, most of which is artificial transcription factors, and then Jeremy Levin, who is representing Ovid on behalf of Downstream Therapeutics and Gabox et al. So I have a bunch of questions that I'm going to speak to because I think it'll be much faster than asking the audience. And we got all these questions in a Facebook survey, and so I wrote them all down. And so those that are listening online, hopefully I have addressed and will address all of your questions. So I will probably just point these toward individuals at the table unless somebody feels very strongly they want to answer it, but just to keep it simple. And feel free to also, if someone else has a comment, to jump right on in it and answer the question. So one question was, what do you think a cure or treatments look like for different age groups of children with Angelman syndrome, knowing there's a lot of talk about therapeutic windows and what opportunities are we going to have for children of various different age groups? And I'd like Dr. Weeber, if you don't mind, to answer that question. I know you always get that question, but you've become so good at answering it that I think it makes the most sense for you to answer that question.
Yeah, thanks.
I know it's a tough one.
And actually, when you were reading that question, I was thinking this would be a perfect question for an MD and not a PhD. I think we're still trying to figure that out. I think that Art's talk, kind of addressed some of that. But I'd like to hear what Art would like to say about that.
Dr. Beaudet, would you mind?
I guess I would think that the younger the age at the starting treatment, the better. I mean, day 2 of life would be a good time. But on the other hand, there's, I think, reasonable expectations that there will be benefit for patients. I would think still huge benefit for patients who are 2, 3, and 4 years old. I think I've had some discussions with people. I mean, if you have a 20-year-old with Angelman syndrome and you treat them, I wouldn't be shocked if they develop some words and develop some speech, but we won't know until it's done. So I think there's likely to be benefit at any age, but the younger the patient, the greater the benefit. I think that age would be a more important variable than which genotype they have, whether they're deletion or something else.
Yes, Dr. Segal?
May I respond?
Yes. I should have known that you'd have a good response.
Well, I think it depends on how you look at this drug development program. So if you look at it as like, are we just going to make these drugs, see what they do, and then we're done? We've done everything we can do. Then that's a very important question. If we look at it as a process, like how do we cure Angelman syndrome? We have these set of drugs. We're going to test them. We're going to see what they do. And for the things that they don't do, we're going to have to find something for those. And so if it's more effective in younger kids, and we still need to treat older kids, then that's the next goal. If it cures seizures, but it doesn't cure cognitive functions, then that's the next goal. So, I mean, whatever the challenges that come up, we just have to address it.
That's great. And And I think it's also important, because we had a lot of those questions, for everyone to remember that it's not necessarily just one treatment. So one treatment may affect one area, one focus, one domain of issue, but another treatment may affect a different one. So it's going to likely be a cocktail to affect everything, but there's no reason to believe at any age that we're not going to see major therapeutic benefit. And so I think until we're in a human, we just don't know the answer to that question. So we can surmise all we want, but we don't know. The next question, which is really about genotypes, so I think we've already answered it, is have we, but I think it is a good question in that, have we used any animal testing to differentiate the genotypes in terms of best therapeutic approach for the different genotypes? So Scott, do you want to answer that one as a geneticist?
So what was the question again?
It was about genotypes. So have we looked in animal models at different genotypes in order to look at best therapeutic approach for different genotypes?
You may be more. Well, the
answer's no.
The model, the mouse model that we have is a loss of function mutation. We have which is a point mutation essentially. I mean, you could think of it that way. The rat is a deletion, but just of UB3A. The mouse model that Dr. Beaudet was referring to earlier, the UPD mouse model, actually have that, but it's a very challenging mouse model to work with. It's extremely difficult. But I would say that most of it's involved the one mouse model that we have. I know there's some others that exist, and the work that's being done in Ed and Jill's lab with the deletion with the rat. The pig that was made was the same thing. It'll be the same deletion of UB3A, but what we don't have, I guess, is a model that is similar to the large deletion that you see typically in AS.
Great, thank you very much. This is a question for Meghan and Michelle, either one of you or both of you. People want to know what they can do to get their children ready for human clinical trials, and where can they be most helpful now in order to be able to be set when the name is called that a trial is about to enroll.
So I think that one of the really important things that all families can be involved in is the registry. So I think in terms of starting to contribute data to that, I think is a really good step. I think in terms of, as we were discussing earlier, changing therapies or I think there's a real value to a lot of the adjunctive therapies, physiotherapy, speech and language, occupational, and I think being in a good position to add in a drug treatment should be on top of all of those measures. So I think, if possible, that one good way to prepare for trials is to really engage with all of those supportive measures that can really have a real benefit, and doing that sooner rather than later, so that, as we spoke, when it comes to the time to enroll in a clinical trial, that the standard of care has already been really optimized as much as possible. So I think that that's something that the local clinics can support with getting in place.
Great, thank you. I think this would be a great question for both Jennifer and maybe Jeremy. So I'll start with Jennifer, but from the regulatory perspective, and then Jeremy maybe from the industry perspective as someone who's currently in a phase 1 or phase 2 clinical trial, soon to be phase 3, as I now know. Would participating in one clinical trial preclude from participating in another clinical trial? And what would be a scenario where it would, and what would be a scenario where it wouldn't?
I think this came up in one of the talks earlier today, is that oftentimes when we're looking at an investigational medicine and we don't know how it's going to behave, we only have data from animal studies, we exclude patients that have been in a clinical trial where they've received another drug because we're not sure if it's safe or if there are carryover effects. So then it's hard to assess if something good happens as a result of drug A plus drug B, or if there's a safety issue, is it that result of that same combination? So generally speaking, you don't see most trials designed where you can be on multiple investigational therapies. You can be in observational studies though, so if you're in a natural history study or a registry, and you didn't receive any therapy as part of that, obviously, then you could get into a clinical trial of, if you meet the eligibility criteria, I will say, as a regulatory person, for that investigational study.
Great, and for Jeremy's purpose, if you're taking a drug like gaboxetol or OV101, and it's a downstream therapeutic that has a washout period, the question was how long of a washout period would be necessary in order to consider an upstream therapeutic like an ASO or a gene therapy?
Yeah. Hello. I think you should step back 1 second. That question that you're posing presupposes a couple of things that are important to consider, and David touched on them, and we've talked about them in the first couple of questions. The point about a cure, it's a one and done, and you don't ever have to see the doctor again. And there's only Jim who's talked about that, really, because a gene therapy is exactly that. ASO therapies, oral therapies, which affect different levels of the disease. And if you have to keep on taking the drug, it's not a cure. You're fixing something. So let's just start with that. And it's an important concept to really internalize, because when you think about that, then your trials start to think very differently. The fact of the matter is that each one of these upstream and downstreams will probably contribute different things. So what one has to construct is the idea that how are you going to be able to work together in the clinical trials to ensure that no matter which drug you're on, that you have a combination eventually. Because it doesn't matter which one comes first. Let's assume that right now we're entering phase 3. That gives the likelihood, not the certainty, but the likelihood that this will be the first medicine to treat patients with Angelman's, which is a good thing. However, one of the other therapeutics may supplant it. Once you've got that approved, all other patients, all other trials should have the right to either do it as a combination or individually. And people may say, look, I'm willing not to take this while I do the other trial. So it's a very, very specific set of steps that take place. But I think we as a community need to be prepared for the probability that a combination therapy is likely to be the best way. So we have to figure out, so I'm coming back now to your question, which is, there is no question of a washout period. You should not be mixing two investigational drugs. That's for sure you shouldn't be doing. However, once one is approved, or during the period of time that you can go to the FDA and request a certain set of preclinical studies to assess whether or not you have a good synergy, and it's a very good idea to test two things together preclinically so that you're prepared when there is a clinical effect to actually say, I have one approved and the other one is coming along. By the way, that's the norm in nearly every other disorder. And we're finding that you don't cure diseases, you actually have to combine things unless you give them an antibiotic or a gene therapy, a true gene therapy.
Thank you. So this is a question for Dr. Wilson. Would a human develop immunity to a virus like AAV? So is it really truly one shot, or do we feel like when newer versions come up that might be better options in the following years, that there would be more opportunity?
The virus is viewed as foreign. When injected into the blood, or really into the thecal space in the cerebrospinal fluid, the patient will develop antibodies against it. In fact, some of us have antibodies against AAV9, and probably about 25% of you, and your children do as well. So the antibodies that pre-exist because of some natural infection that you had or an initial treatment could interfere with a second, or could preclude you from participating in a trial. That's if the vector is injected into the blood. But pre-existing antibodies that exist in populations did not interfere with the efficacy or safety of a treatment when it's injected into the cerebrospinal fluid. So it's one of the advantages.
Great, thank you. The other question to follow that, which you probably also could answer, is would there be an opportunity if there was concern for an immunity if different viruses down the line had different opportunities? If you had, for example, an AAV virus and then 5 years later a lentivirus, would there be as much concern about an immune reaction if you're using two different viruses?
No, generally they don't cross-interfere with one another.
These are all questions from Facebook. This was for Dr. Anderson about cord blood, so we've already answered that. I think we've answered almost everything here. Yeah, everything here has been answered throughout the course of the day. So why don't we take another couple minutes and just have people in the room that may have more questions than what I have already asked, but almost everything was already asked earlier.
Thank you very much. I have a question from today and last year. I remember that Dr. Beaudet said that we have some people have some level of paternal expression of UB3A, and that there are people that can be completely normal. They have no like AS symptoms, I think I remember that, but they have so much level of UB3A paternal expression that we don't notice that they have the disease. Is my memory wrong, or I did something that was resemble?
I'm having a hard time trying to put it in context and recollect exactly what was said. It is possible or probable that the extent to which the father's copy is shut off may vary from individual to individual, so that maybe it's 99% shut off or 95%
Okay.
or something like that. So there may be some leaky expression from the father's copy of the gene. We don't have very good information about that, but when you look in the mice and you have knocked out the maternal copy, you can often still see some UBE3A there. Now, whether it's coming from other cell types like glia, or whether it's it would have to come from the paternal copy, and so I think there's still fine points there that need further elucidation.
Okay, so there's nobody that has like 80% of expression of paternal allele.
The mosaics, I think maybe it was the mosaics that you.
Oh, so Scott's mentioning maybe it's mosaic. Now, there are individuals who have Angelman abnormalities in some of their cells in their body, and then they have other parts of the cells in their body that are normal. So these are mosaic individuals. There, the percentage could be anything. It could be, 90% Angelman and 10% normal, or it could be 50-50. That's a different situation with mosaicism.
Okay, thank you.
So my question is for Dr. Timothy Yu, I guess. We, in the mutation group that I follow on Facebook, and my daughter also deals with it, we're dealing with they lose the ability to walk, or the ability to walk better or whatever. And I was curious if our doctor said that maybe something else is causing it, since we don't know everything that a mutation kid had. So I was wondering if the RNA thing that you did, would that be something that we needed to look into to see if there was something else causing that, since I don't know if that makes any sense, but.
So let me just try to make sure that I understood your question correctly. So that there are some additional problems that folks in your mutation group are experiencing.
Yes.
That are above and beyond what other genotypes
Yes.
experience. Right. And may I ask, are we talking about a contiguous deletion group compared to the point mutation group, or are we talking about?
The UPDA, but UB3A mutation is what we have.
I think she has a mutation Facebook group of kids that just have the genotype mutation, and they're losing the ability to walk as they're getting older. And she's trying to understand if losing that ability to walk is another genetic abnormality, or if that is associated with the mutation itself. Is that correct?
Yes, ma'am, sorry.
And I'd welcome any input from my colleagues here. What can I offer? I think that one thing that has come up, and Art, you know this very well, is that occasionally lightning strikes twice.
Okay.
And sometimes what we think when a child presents with a neurologic disorder, and we find one smoking gun in about 5 or 6% of cases. There's actually a second hit someplace else at the genome that's happens to be there. And so it takes a real experienced clinician to make sure that we're not closing the diagnostic book too soon. So that's one potential answer. I think that's only 5 or 6% of cases, but it is sometimes the case. I was going to say one thing. I think that you mentioned that within specific mutations, are there places where we ought to think about the same sort of RNA therapy that I discussed? Scott mentioned that there is an RNA, that we're talking about using the same sorts of oligonucleotides. The chemical structures are similar. The particular strategy that genetics and Scott are advancing is a little different than Melicin. There are opportunities potentially for patients with very specific types of mutations to consider a drug like Melicin. If you have the right type of mutation in the UBE3A gene, it's possible that out there somewhere in the world there is a patient with a UBE3A mutation that has the same characteristics as what we found in our patients. That is theoretically possible. I don't want to distract from the important work that's going on through genetics that can help the whole community. But since you raised it, I wanted to mention that possibility.
Maybe I could just ask a question more than make a comment. Yesterday, I heard mention of something that was new to me of non-eleptogenic myoclonus. And I think I tried to look up that term in PubMed this morning, and you don't get any hits. And if you put it with Angelman, you certainly don't get any hits. So, and I guess it would be, could that be a factor, and what would be the age of these individuals who are having more trouble walking than they used to?
That's exactly what I was going to say. Is my first recommendation was that not to make Ann Anderson have a lot of work to do, but she is a clinician, an epileptologist and a neurologist who sees patients of Angelman in a large number, and so she can probably talk to you a lot more to this. But what has been noted, and potentially even more so, I believe, in a mutation genotype, but in all genotypes, is that there is something called non-epileptic myoclonus that happens as kids get older. And it happens in over 50% of kids over the age of 13, and it happens more over 17. And so that is something that is a possibility, but we can talk about that separately. I don't think it's really for this panel, but that can affect their ability to walk. Usually they have almost like convulsions, but they're not seizures, or not thought to be seizures. But that can affect their ability to walk, as well as scoliosis and other orthopedic hip issues and things like that. And I think if it's happening in a large number of mutation kids, it's not just one individual, it's unlikely that it's a second lightning strike. It's more likely that it has something to do with the growth of the bones or the muscles or contracture, or something that is Angelman related in that type of group, is what I would guess as a veterinary clinician. But probably for another time.
I just want to say, Allison, I just want to interrupt for a minute, because if non-epileptic myoclonus is very hard to miss. It is not subtle. If I don't think that would be it. If it's just a matter of mobility deteriorating, I wouldn't automatically jump to non-epileptic myoclonus. That is debilitating and changes the child's entire quality of life, not just with declined mobility. So don't panic that that's what your child has, because I think you would know there was a lot more wrong than just a deterioration of gross motor.
There's also a paper on adolescents and adults in Angelman syndrome published recently by Rna Therapy that talks about, as some groups of children are getting older, they are potentially losing the ability to walk for various reasons, most of which is orthopedic. So it's something that you should look into as well, because that may be similar to, you can read about the symptoms and see if that's similar for you. Other questions?
Yep.
Hello, my name is Charlotte and I'm coming from France. So since there's a lot of stream that is hopefully getting into clinical very soon, and hopefully with a treatment one day approved by FDA. So I was wondering whether for us in Europe, what would be the next step for having this treatment in Europe, and how can we help you on this?
Jennifer, this might be a good question for you on the regulatory side.
The process in Europe is actually very similar. You file what's called a clinical trial application. And oftentimes, I can't speak for all the companies and plans, but oftentimes what happens, at least in my experience with rare diseases, is we try to do a study globally because there's not that many patients, and so it helps us recruit the study faster. And so what you do in Europe is you approach it at the country level, and you file a very similar clinical trial application within that country, and you go through a similar regulatory process. Not exactly the same in terms of timelines, but generally the information that you're providing to justify taking the study forward is quite similar.
Just a comment on that. We have actually started that process in Europe. We have run a clinical trial which is global. One of the sites was in Israel. And so our approach is that all patients, and I think FAST says there's 500,000 individuals around the world who should be addressed, and we're not going to leave any of them behind.
Comment as well on that, and I think another thing is around basically accessibility to patients in Europe, and I think being involved in the patient communities in Europe as well, like the Angelman Syndrome Alliance or the specific country patient advocacy groups, will be very helpful, because as a company we start to reach out to these groups and to understand how to access and approach patients in different countries.
And the global registry. So with the global registry, we can see where you're from and see where the highest populations are. And that might be an impetus for companies to want to start a trial in that country because they know that there are a lot of individuals in that country. So another shout out to register for the registry regardless of what country you're from.
I have a kind of a series of questions related to the first question that was asked by Allison, which is related to like the age of the treatments. So is there like a focus age? Like when we start trials, are we going to start with 18-month-olds, 6-year-olds, 20-year-olds? Is there like been any thought to that as to where it's going to start? And then based on that, thoughts on CBD oil for treatment of seizures and how that CBD oil use will affect getting into clinical trials, because we've kind of heard that that will be the kind of affect their ability to get into the trials.
Dr. Wilson.
So our experience with a more risky approach that's never been tested in patients with that disease is, at least on the FDA side, they push us really hard to initiate in adults. Even though they can't consent. And so I would expect that would be the case. Whether that would be appropriate or justified would it would be based on the data at the time. But the first I'm pretty sure if we were involved, we'd be pushed to enroll adults. But then that's a small trial, and then you can leverage that to expand into larger trials, and that maybe I tell you it's different than in Europe, our experience, where they're more open to a broader range of ages for the first trial, and then it's a whole clinical development plan. You got to start somewhere and then you expand. And colleagues at Roche and Jeremy could probably comment on that.
Yeah, doesn't SMA present some precedent in that regard?
Yeah, but it's a different disease.
Yeah, I think SMA is not a good example.
No.
I think SMA is one where you see mortality happening. It's a severe disorder, and by golly, you don't have much choices. And the difference here is that you have individuals who live many, many years. And our experience, though, to your point, Jim, is that we were asked by the FDA to start in adults, to demonstrate safety, to demonstrate effectiveness, and then, and as you've seen, we've only now launched into the little guys. And I think it's an approach which will probably, I don't know if that's the way they'll adopt that for the approaches that have been adopted to adjust the the types of therapies that we're discussing here on this side of the table.
Sorry, I just wanted to add. I agree with everything that's been said, and generally speaking, FDA wants you to start in adults because adding on the pediatric piece is another sort of complicating factor because you have children that are developing and you don't know if you could somehow impact that. I can say at genetics, what we're trying to do is actually start a study in pediatric patients. And so it's something that we're trying to carefully approach in terms of how we study the drug in animals to make sure that we have support to try to go into pediatrics as early as possible.
I would think that the families would want to convince the FDA that going into young children shouldn't be delayed extraordinarily. I mean, it's one thing to delay it by 6 months, but to delay it by 6 years would be unconscionable and not ethical.
That was, sorry, thank you. That was,
Did you have one more question, one more answer, Jim? Did you want to say something?
Question for the panel.
Oh,
Since
about
they're
this?
all here.
We'll do CBD after. If you
The
have
CBD.
Yeah, I just think
Yes.
this is on the same topic, then Jim can ask it. So for CBD, I'm not sure that anyone on the panel, but Tim probably can answer that best as the clinician who's seeing patients most regularly.
Well, I'm not sure I want to, we haven't discussed inclusion criteria with respect
Yeah,
to
I don't think we
CBD,
can
so
speak
I
to
think
the
it's
trial
hard to say.
itself, but just in general to your experience with clinical trials. I can speak to my understanding with clinical trials, and certainly with a daughter who's on CBD. Our understanding, or my understanding from the various different companies that we have talked to and the various different information that we have gotten is that it depends on what drug it is, whether it is something that is looking at similar things in terms of endpoints to what CBD is doing versus if it is something very different. So it really depends on the company, the drug, the mechanism of action, and the stability of what you're on. So if you're on CBD, for example, and you're looking for sleep, and you started on CBD for a year, and then you're looking at sleep for 6 months, and you're on the same dose of CBD for the following year, then that would be acceptable. But if you're looking at anxiety and you only started your CBD a month ago, that would be unacceptable. So I think it's very specific, is my understanding, and it's very specific to the drug itself and to what you're trying to learn from that drug. I hope that's helpful.
And Allison, wasn't Epidiolex just FDA approved? So Epidiolex is a cannabidiol anti-epileptic. It wasn't approved for Angelman, obviously, but is now an FDA-approved drug. So if you're on an FDA-approved drug, then it's no longer considered experimental. So for those of you that really want the benefits of that type of therapeutic, you should speak to your neurologists about maybe trialing Epidiolex on your kiddos.
Yeah, and it's now apparently covered by most insurance companies, as far as I've been told. So you can talk to your neurologist about that. Question?
All right, mine was regarding the paternal allele. Is there evidence that the paternal allele will be able to do what the maternal does? And I guess my question more or less was coming, okay, as the mother giving a chromosome to my child, it could have been the one I got from my father, and it could have been the one I got from my mother. Am I correct in that? So potentially I could be passing on one that was my paternal one versus my maternal one? Am I understanding
do you
that
wanna go for
correct?
that?
Just clarify.
I think he gets what you're trying to say.
Yes, so the imprinting process resets everything with each new child. So even if it came from your father or your mother, it's going to be set as maternal when you transmit it. So the system will have to reset your father's chromosome, but just kind of keep your mother's chromosome as it is.
So is that evidence then that the paternal chromosome could do the job? I mean, is that part of the research?
I think we know if you transmitted it through to a new patient, it could become maternal and do the job. Whether if someone activates it in some way, whether it will behave exactly as a maternal chromosome would behave is an important variable in how well the whole thing might work. But we do know that it looks like the early part of the paternal copy of the gene is being transcribed and made into RNA and may well be under the same controls that the maternal would be. So I think that's an issue that's critical but likely to be favorable.
I think, Dr. Wilson, you had a question for the panel.
Yeah, it's it's a challenge that we encounter at the Orphan Disease Center, where we're involved in a number of rare diseases for which there are no approved therapies or treatments. And we aspire to these observational natural history studies under protocol where you establish a trajectory of the disease and characterize the disease so that when you do have a clinical trial, you can use that as a comparator group. But what we're seeing as we succeed, such as hopefully Jeremy will, that the standard of care changes. And as the standard of care changes and we're successful in bringing new drugs in, those natural history studies, observational studies become obsolete because the new comparator group is on top of standard of care. So I guess that would be a high-class problem, but it is one just to be aware of as the whole field sort of moves forward in parallel or in aggregate with some of these studies or observation.
Great point.
I'll echo Alison here a little bit, and I guess our intention is really to be able to move forward, I think all of us at the table, at a rate where we're coming to clinic before there's an opportunity for things to evolve to such a state, as for example in Duchenne muscular dystrophy, where the standard of care did change significantly through the broad use of steroids and how that impacted the disease progression. So I think if we all move at the speed that I think we're all speaking to today, that we should be able to actually be in clinic and use these data before the standard of care has evolved and settled to that extent. But I'm being hopeful here, so yes, be positive.
Optimistic. We're going with optimism.
Exactly.
It's the way we roll around here. Okay, one more question. Last question.
First off, I just want to thank all of you for everything that you do. We have a 3-year-old with a deletion, and we fight constantly with seizures. My wife and I have pretty much done everything possible to control his seizures, and he's still having seizures. So you guys give us the hope to keep fighting and keep doing what we need to do on a daily basis to help our son. So we want to thank you for that. Now, on to something positive. We're looking into the future. We had this question, we were pondering it. Now, once something's developed, like implanting the gene, etc., what's going to happen as far as getting insurance to cover it, the cost to the parents, etc.? I mean, I know, Dr. You've been through this. How does that work with insurance companies once we get to that point? And where do we go from there?
I know who really wants to answer this question. So do you want to take this, Jeremy?
Oh, yeah. Look, what's absolutely wonderful is the series of absolutely fabulous new therapeutics coming along here. You can hear it. We heard all about it this afternoon. So each one of these is going to require the fact to deal with the insurance companies. You should all know that 5 companies today cover 140 million Americans. Those 5 companies are trying to figure out right now, and by the way, it could get smaller than that. Smaller numbers covering more people. So we're in a different situation in this country than we were 3 years ago and 5 years ago. Second item that's changing real fast is how they think about and are willing to address the costs of different types of medicine. And it's them who are going to determine this. So the address that every single company here will have to go to is one of 4 places. There are maybe 5, maximally. So about 4 years ago when we started Allison, we actually brought somebody in on day 1 to ask how would we be reimbursed? So it's a process which has evolved even since we started. But I think that each one of us has a responsibility to you to ensure that by the time that the therapies are available, whether it be a genetic therapy, whether it be a gene therapy, which is very different, lots of contemplate, how do you spend the money on a gene therapy when you're going to do it once and then it's done? How do you deal with that? Nobody has yet come up with a good answer for that. Amit passed over this. People started to tackle this in cancer. It's not a good model because those people in generally demise. What you're dealing with now is one therapy for their lifetime treatment or multiple therapies, 1 or 2 or 3 a year, all of which, if we use, I don't want to use Spinraza because it's a terribly expensive example. It's $750,000 a year. That can't be. No insurance company is going to agree to that. And I'm not sure how we deal with the chronic, long-living ones. So what I'm trying to say to you is that we as a group, including yourselves, need to tackle this with the insurance companies now. This is not an issue that should be ignored. And we didn't ignore it to begin with. We know what we're going to do with it. And we'll be happy to share our experience with many of the others. One thing you can't do is you have 3 different approaches here on ASOs. You can't collaborate on pricing. Okay? You should understand that. It cannot happen. They can't even talk to each other. It's illegal. And so as we start to talk to the insurance companies, we have to recognize, Alison and others who are in this, that with that type of an approach, the world of pricing and those who control pricing is going to be completely different in 3 to 4 years than it is today, and that's completely different from what it was 4 years ago. So I'm very conscious of it. I spend a lot of time on it. I don't have good answers for that end of the equation. I hope I've got the answers on our end of the equation.
Doctor, do you want anything to add?
I agree with everything that Jeremy said, both in sentiment and in practice. It's a problem that still needs to be worked out. I'll offer one additional perspective, which is that in addition to thinking about traditional drug pricing models, there are other types of treatments that we offer to patients, not necessarily as drug companies, but as physicians or as hospitals. I'm talking about transplants. I'm talking about a bone marrow transplant. Or organ transplants, or dialysis. Right? What's distinguishing about these kinds of treatments is that they aren't drugs. They don't improve and also what's distinguishing about them is that they don't improve your life a little bit. They improve your life a lot, but they're very, very expensive. And the kinds of interventions that we're hoping to achieve, all of us at this table, we aspire to have that same sort of impact, right? Cure, whether or not you like the word or not, to actually have really significant impact with big, big changes in improving the quality of life of kids with Angelman syndrome. That's all what we aspire to. And the methods by which society has come up to appropriately set reimbursement for life-saving, quality of life altering treatments like that have involved public policy, advocacy, organization, not just at the market force level, but also just real engagement. People vote on this stuff. So I'm just seeding that because that's another parallel pricing discussion, and one that I hope that people think about too and figure out ways to get ahead of the game in thinking about how to deliver these at scale.
And just as you at FAST and the families around the table here have driven the interest in finding medicines that can be helpful, so I'm afraid, and I advocate that you do get involved in this aspect, because you're right, people vote on this stuff, and there's a lot to be done by the organizations to drive decisions within these insurance companies and payers.
Thank you very much. And I'll just say, and I know Paul is probably dying to say something, but I'm going to put my FAST hat on and tell you that as FAST, and I'm sure ASF feels exactly the same way as foundations and organizations, we are 100% on board to advocate in any lobbying political format that we need to. As parents, that pricing will not hold any child, as far as we're concerned in this world, from getting treated. And that is a commitment that we have made as a foundation.
And I'm just going to add to that. You guys are struggling every single day to get your child's seizures under control. You should not be worried. about the pricing of the drugs that are going to help your child, or your insurance companies approving them. You have enough on your plate. You know, we're addressing it. We're all very conscious of it. Don't feel like this is your burden to bear, you know?
So I would like to end there. And did you have something? You raised your hand? Okay. It was 2 or 3 years ago. It was Ovid that presented from London School of Economics the cost of care, and there was going to be a study that was looking at the cost of care in the UK, I think. Was that right?
For the insurance companies to see if you do a single treatment and you save a lifetime of what the insurance company would be paying for therapies, that tends to balance it off and convince them that maybe it's a worthwhile investment. So I don't know where that study stands or if it ever came to fruition.
The study is very rich. We have a huge amount of data on that. So this deals with what is the cost of every aspect of an individual's life. How much are you losing by not working? When you don't sleep at night and you can't go to work next day, what happens? So we don't just count what is it the doctor's visit. It's how much does it cost then for a nanny, somebody else to come in, a caregiver to supplant you. So these costs, we have a very good series of estimates in this, and they are very substantial. So that when the right time comes and FAST and ASF are going to engage in that, I think that's, and I hope some of the larger companies will do this as well, that we'll share what this is, and we'll have a very good set of studies and good analysis of it. But we've been doing this for 3 years in preparation exactly for this moment.
Great. So I'm going to round it up there, and I want to thank our panel discussants for being here and for being so supportive of the Angelman community. And I think, as you can see, the parents are so grateful to every one of you for being here and for caring enough about our kids to make a difference. So we thank you all very much.